Coming to a case study, I take the example of sugarcane, which is of high biomass producer. A well-developed cropping and downstream processing technology is available with respect to sugarcane, and the standard protocols available for genetic transformation. Biosafety issues are relatively less with sugarcane because commercially grown sugarcane is vegetatively propagated and the sexual seeds are not used for commercial cultivation at all. It is not flower in many parts of the country, and even if it is flowers, it does not set the seeds. And no reports of pollen transfer from cultivated sugarcane to its wild relatives is recorded, hence no chance of transient introgression to wild Saccharum species. No weediness observed as sugarcane is vegetatively propagated and requires ideal conditions for establishment and growth. Biomass partitioning in sugarcane, you see the root part is 10%, and then 70% of sugarcane biomass is of economic importance, that is the stem part, which gives 70% of biomass, and then 20% of the leafy part. Top of the sugarcane is leaf, and then these 70% of stem part has maximum space occupied by the vacuole where sugar is stored. So when we transform the sugarcane by targeting a particular protein to the vacuole, is going to be a superb success because of the high excess of the scalability or excess production of the protein molecule in the vacuole. And then identification of targeting vacuolar targeting sequence and validation. So with the basic idea to target the protein to the vacuole, we started looking for a targeting sequence or signal peptide, so that we can attach this signal peptide to a gene of interest, and then gene of interest will be expressed in the vacuole. That was the idea. We have based on our hypothesis, we have identified a signal peptide of 78 nucleotide in size, and then we have made a different deletion of these 78 nucleotides. This is identified based on the gene bank sequences, and then we have prepared a different construct or plant expression construct, and then we identified we have fused that targeting sequence with the protein, and then we transform this protein construct to the sugarcane transformation, and then we characterized its expression in vacuole through GFE localization cells by staining procedure. And also after transformation, we have extracted the juice, and then we have performed the SDS page analysis to verify quantify the protein expressed in the sugarcane juice is of the size equal to the commercially available. That way we have confirmed our gene of interest should express 70 kDa Dalton protein. That is confirmed through SDS page analysis. And then this is a simplified scheme which I explained previously, and then this is the best way to get sugarcane product in sugarcane. And these are some of the antibodies produced in the transgenic plants, and then some of them are produced like edible vaccine, and then hepatitis B antigen, and then there are numerous proteins or the non-protein or non-pharmaceutical proteins, or the pharmaceutical proteins are produced in the plant system. These are the plant derived oral vaccines produced in related to biosafety issues in molecular farming. Gene and protein pollution, transgene and their encoder proteins could spread into the environment and could affect non-transgenic organisms. Ultimately, humans could be affected by the consumption of food containing such genes or recombinant protein. These are one of the risk, the issues for the public concern. And then the product safety, such as pharmaceutical products, and concerns the risk that such products could be harmful in human or animal patients to which they are administered. Some of them are These are the different concerns, and then opportunities for purified products. These are the different products we available commercially, like Avidin and gas, the biocatalyst like trypsin, biopolymers, that also produced here, and then some of the other products like collagen, keratin, and silk, elastin. These also be produced in the plant system. And then feed additives. Not only directly we can use it for human consumption or the pharmaceutical purpose or the non-pharmaceutical purpose. Here you can see the feed additives. So example, the anti-nutritional factor like those are K to remove anti-nutritional factor, but like phytases are required to remove the anti-nutritional factors. And then the enzymes related to fiber degradation, like hemicellulases, which helps in digestion of the fibers that can be produced. And then the enzymes beta mannose and beta glucanase are often added to animal feeds to eliminate anti-nutritional factors. In this case, the targets are mannose and glucans, which absorb water and increase the viscosity of scheme, thus restricting access to the intestinal surface and reducing the efficiency of nutrient uptake. These are the important things. And then with respect to the biofuel point of view, the biofuels derived from cellulose and hemicellulose would therefore be more sustainable and would not compete with the food and feed. So we need to have some of the enzymes produced in the plant system for the sustained production of the biofuels. And then another point is the paper making or paper manufacturing. In the paper manufacturing industry, the raw wood pulp undergoes a delignification process to separate the lignin from the cellulose fibers, and the cellulose rich pulp is often bleached to produce white paper. Both processes require harsh physical and chemical treatments that generate environmental pollution. Lacase is one of the enzymes could also be used to treat textile mill effluents, remove phenolic compounds from the beverages, remove sulfur from fossil fuels, and develop biosensors. Plants that are engineered to secrete lacase from their roots could also be used to remove pesticides and xenohormones from the soil. Some of the industrial enzymes are produced, then different applications are indicated in this table, like alpha amylase, tobacco plants, food processing, Avidin, maize plant or corn plant in diagnostic kits, cellulase, brassica plants, production of alcohol from cellulose, that is for biofuel production, beta glucanase in brassica in brewing industry, beta glucuronidase, brassica in diagnostic kits, lignin peroxidase, wheat in paper manufacturing, phytase, rice, improved phosphate utilization, trypsin, maize, pharmaceutical purpose, xylanase, brassica, biomass processing, paper and industries. Trypsin, pharmaceutical purpose, gus, it's a reagent for diagnostics, Avidin, immunological reagent, approteinin, wound closure, collagen, gel cap, lipase, exocrine pancreatic insufficiency, lactoferrin, natural defense, TGEV, edible vaccines. Some of the non-pharmaceutical proteins are produced. In summary, the plants are effective and efficient bioreactors for the production of pharmaceutically valuable recombinant proteins. 2. Variety of plant species that are being explored to serve as a green bioreactors. We have variety of plant species which can be explored to serve as a green bioreactors as per our requirement. And then transgenic plants shows low production cost, high productivity, low risk of contamination and easy storage compared to transgenic animal. The fourth point is the plant-based production systems are established on the market. The first product for human medical use was commercialized in 2012, but currently the non-pharmaceutical products outnumber the pharmaceutical products comprises research grade or diagnostic reagents, cosmetic ingredients, biosensors or biocatalysts, but also industrial enzymes used in the feed, biofuel and paper making industries. The point 6 number, key advantage of plant derived products are animal and endotoxin free production compared to the animal system. You have the endotoxin production. Then when we produce it in the plant, it is endotoxin free production system. The cost advantages for the raw materials and the process is low with the plant based production. Large scale use of technical reagents, antibodies and industrial applications could become feasible, but scale up will require some investments to install equipment for these products. 8th, plants are renewable biomass for energy production. So it's a renewable source, it's a sustainable source. Enzymes used in this biomass conversion process could be produced directly in the plant biomass rather than in separate migrable systems. You see, plants are renewable biomass for energy production. Not only that, the enzymes used in the biomass conversion process could also be produced within the plant system. Then, rather than producing it in another system and then bringing it and applying it, the processing with the plant biomass is a cumbersome and then expensive. 9. Further development and widespread success of the technology will be strongly influenced by the level of regulation and restrictions applied to genetically modified plants and products derived thereafter. In order to directly benefit from the technology, countries will have to be proactive in the regulatory process. With this, I conclude my lecture. I am ready to take any queries. Thank you very much.